Thank you. It's an absolute honor to be asked to give this lecture. Chuck showed me the previous 10, and you go, Jesus. So I appreciate that. As far as the quality-quantity thing is concerned, I'm reminded of a New Yorker cartoon where a woman goes into a speed reading class and is given 3 options. She can learn to read faster and slightly diminish retention. She can learn to read much faster with a compromise of retention, or she can forget retention altogether and just read very quickly. And so the quality-quantity thing sort of reminded me of that. It's an honor to be here because we share so many different objectives in the context of development. And clearly one of the objectives that we change, particularly in the issue of human development, is the question of how early experience is embedded in the biology of an individual and how it comes to so profoundly influence health over the course of the lifespan. By the way, I'm going to wander around so that I hope you guys can see slides, because I think I'm standing between you and that show, and that's more important. So what I want to suggest is a possibility that, at least in part, this biological embedding, as Clyde Hertzman would phrase it, occurs at the level of the DNA. And it occurs at a level that is functional with respect to transcription and brain development. And indeed, it defines, I think, a process by which we can come to think of the social environment as being imprinted directly onto the genome. And because these effects are potentially chemically stable, the imprint of childhood adversity can be seen to be a mechanism that might explain the enduring effects on health and capacity over the lifespan. So here's where we begin. So this is just a quick synopsis, if you will. That's something you know all too well. This is really just the results of about 15 years of research in social epidemiology. And what it describes is simply the statistical relationship between the quality of early family life and the probability of developing any one of these particular disorders. And the interesting thing to keep in mind is that this is not unique to mental health, that you can see, in fact, the implications for metabolic as well as cardiovascular disease. And I think the interesting subtext to that is that it also provides us a mechanism for understanding how it is that comorbidities may emerge in relation to common environmental events. And I think that in many cases, we've kind of missed the boat in human development because many of our studies, particularly intervention studies, have focused so much on brain-based mechanisms that they've ignored the possibility that these same mechanisms targeting the same environmental conditions might have impact at this particular level. And I think it was particularly gratifying to see James Heckman's group go back over and analyze some of the abecedarian work and show, in fact, impact of the Ramey-induced interventions on the risk for all-cause mortality. So I think what we need to do is to think of this as a system by which multiple features of biology are being effective, associated with many health outcomes. So one of the questions, of course, is that a pathway? How is it that the quality of early family life might be imposing changes on the development of functional systems which then mediate vulnerability over the course of the lifespan? And what you're looking at here is one of what is referred to sometimes as a stress diathesis model. It's sort of a working hypothesis in biological psychiatry. And what it suggests is that early family life influences the development of individual differences in endocrine neural response systems, and that these systems and subsequently those individual differences are then predictive, if not causally related to these outcomes. So this is a working model, right? It's just a hypothesis, but it has a certain amount of support. There's recent studies, particularly in humans, suggesting that there is, in fact, at least a statistical association between the quality of early experience and the nature of an individual's responses to stress in later life. And of course, the hypothesis is buttressed by years of studies in clinical medicine showing that the hormones that we produce during periods of stress, cytokines, various neuropeptides, glucocorticoids, catecholamines, can all actively promote each of these diseases, including, of course, diabetes and heart disease. So it's a working model, but one with a certain degree of internal consistency. But the question for us really was, how would this happen? How is it that variation in an early family condition might engender a developmental outcome that persists over the course of the lifespan. How does that actually occur? And so to bring it down, the real question is how do environmental signals act over development to stably affect the activity of genes that regulate physiology and behavior? And we've come to look at epigenetic mechanisms as a candidate here. Now, for those of you unfamiliar with epigenetics, I tell you that it's been with us for many years, right? And people have been studying epigenetics since the 50s and 60s. But it's only somewhat recent that its biochemical basis has come to be fully appreciated. And so what we've done essentially is to start trying to ask questions about whether these epigenetic mechanisms could be extended to understand the development of individual differences in the function of the brain and other organs. Now, I'll give you an idea of where epigenetics has been used in biology to understand fundamental paradoxes in development. I was sharing with some of the people my background in developmental biology, but the interesting thing about developmental biology is that it embodies what is really one of the great paradoxes in biology, and that is, how is it from a common DNA template we produce over 220 different cell types? And every cell in your body has the same DNA. So you produce brain cells, muscle cells, et cetera, et cetera. How do you do that? And the way you do it is by altering the activity of the genome in a tissue-specific manner. So yes, every cell has exactly the same DNA, has the same transcriptional potential, but that potential is regulated by an epigenome. And the epigenome we can think of acts like switches. What it does at any particular site is to determine the level of activity of a specific gene. And it can do so in tissue-specific manners. And one of the classic signals is that of DNA methylation. This is certainly the best studied signal. It's the one that's probably the most chemically stable, I think that's fair to say. And essentially what is thought to occur is that as you add methyl groups onto particular regulatory regions of the DNA, you silence gene expression. And this occurs progressively. Now, I caution you, this story is becoming increasingly complex. So it's true of methylation in one region of the genome may not be true of another. But suffice it to say, when regions that control transcriptional activity, when those regulatory regions, when they bear a methylation signal, on the general effect is to silence gene transcription. So what happens then is that you end up across tissue with the same DNA, but bearing different epigenetic or methylation signals. And that, of course, is then associated with differences in the activity of specific genes. And so that if you look, for example, at the brain cell, it's going to have the same DNA, but it's going to bear a different epigenetic signature. And that epigenetic signature is going to determine why certain genes, for example, show similar activity, whereby other genes show differences in activity. And it's the differences in the genes, of course, that are transcribed that defines the identity of that cell. So a neuron's a neuron because it turns on genes associated with neuronal function, and that's what distinguishes it from a blood cell. So differences in epigenetic signaling, then, is associated with variation in genotype-phenotype relations. Now, we can extend that. We can ask, well, could that be true at the level of the whole organism? So we could start to ask, for example, whether or not things like twin discordance could actually be associated with changes in methylation. And so why might two individuals bearing the same nuclear genes show differences? And it may not surprise you to know that Phil and Fred's mother here doesn't have a whole lot of trouble telling them apart. They've come to differ, and in ways that are more or less charming. And so what is emerging, of course, is an understanding that Phil and Fred share the same nuclear genomes, but that the epigenetic signatures on those genomes, at least in particular tissues, are quite different. And so it's the same matter whereby these epigenetic signals are defining changes in gene activity, thus giving rise to differences in phenotype. So the same can be said when we start looking at adversity. And this is where, of course, I hope to take you, is that essentially when we start looking at early life adversity, that its effects are comparable, that it's actually working to remodel those methylation signals, which then give rise to differences in transcription, and particularly, of course, involving genes that are involved in the regulation of stress responses. And where I'd like to take you a little bit to is also some thinking forward, and that is, if we can understand childhood adversity as a predictor of vulnerability. Recent studies, particularly led by Andrea Denisi at University College London, show that these same signals also seem to be clinically predictive, that they may help us understand why it is that some individuals are more likely to respond to particular treatments than are others. And in this case, the treatment is pharmacological treatments of depression, because what we particularly lack within psychiatry, of course, is the ability to predict treatment outcomes. So it could be that understanding adversity takes us not only to an understanding of vulnerability, but also is clinically relevant in allowing us to predict treatment outcomes. And indeed, I think the hope, of course, is that we might be able to say, well, if adversity lays down a particular epigenetic signal, perhaps we can use that signal to differentially diagnose and treat. So, with that, let me take you to a little bit of the story. So what I want to do is, having given you the broad scope at about 40,000 feet of epigenetics, take you through a little bit of the details of what it is we study and how it is that we study it. So one of the interesting features we were talking earlier with some of the graduate students is that by its very nature, gene transcription is an interaction between the genome and its environment. That's molecularly the nature of gene transcription. Transcription, for the most part, unless you're talking about things like promoterless genes, is regulated by the binding of transcription factors to certain regulatory sites, which in turn turns on the activity of the gene. When you look at transcription factors, these are proteins that exist within cells, and they are chemically activated by signals. And many of those signals involve hormones, drugs, nutrients, etc., that are external to the organism. So in a sense, the activation of the transcription factors that guide gene expression is an environmentally determined event, hence the reason why molecularly gene expression is by definition an interaction of the genome and the environment. Now, that all presupposes, of course, that these transcription factors can easily gain access to DNA, and that's not the case. So here you're looking at the crystallography of Luger and colleagues many years ago, and it defines what really DNA looks like, its living environment. And this is a crystallographic representation of a nucleosome, and it defines chromatin, the interaction between histones and DNA. And what you can see here is that essentially a nucleosome, if you look in an electron microscope, DNA seems like a bead on a string. These is the beads. So it's 146 base pairs wrapped around a histone protein. And the intriguing feature of this is that the positively charged histones, negatively charged DNA, they form a fairly tight complex. And the reason why that's of any interest is that in this particular structure, transcription factors cannot easily gain access to the DNA. So this interaction that I spoke to you earlier about isn't simply a matter of activating transcription factors, it's a matter of allowing those transcription factors to gain access to the DNA. And that's a regulated process. That's the process that's regulated by epigenetics. So one of the simplest epigenetic modifications that occurs is that you take these histone proteins and you modify them. And the most common source of modification are these tails that protrude. These are just amino acid chains, and the tails protrude outside the nucleosome, and they're modified chemically. Acetylation, simplest modification that occurs to histones. When it does, it neutralizes the charge of the histone proteins. Histones open up, transcription factors combine. Many of the factors we identified back in the 1980s as transcriptional cofactors, they're histone acetyltransferases. That's what they do. They acetylate histone proteins. Now, that's what allows the transcription factor to gain access, but this is a transient modification. Histone acetylation, well, we don't have real estimates, but it lasts seconds, maybe a minute or two, but it's clearly an on-off type of process. So it isn't the kind of modification that is going to allow us to explain the enduring effects of any environmental condition. But one that might is the classic modification, and that's DNA methylation. So methylation occurs across the genome. In mammals, it occurs uniquely at cytosines. And it is a modification that I said is associated in many cases with the activity of gene expression. Now, the way in which methylation modifies gene expression is that when the DNA becomes methylated, it attracts a protein, a class of proteins known as methylated DNA binding proteins. Makes sense, right? Methylated DNA binds to it. And what it does is it attracts repressor complexes. These are just clusters of proteins. And one of the main components, in fact, the most active component in these repressor complexes is the presence of a histone deacetylase. What histone deacetylases do is exactly as the name suggests. They kick the acetyl groups off these tails and they prevent reacetylation. So what they're doing is preventing that mechanism that opens up chromatin. And by biasing then towards closed chromatin, you're disfavoring the ability of transcription factors to bind to the site. So that's the process, or major process, by which subtle changes in methylation can silence genes. How's that? Is that okay? Good. If you got that, the rest is simple. In fact, that's not really hard either. If it was, we could never do it. So what we want to then ask is, could these methylation signals, could they be altered by environmental signals working through intracellular machineries? And so we decided we wanted to ask this question in the context of what we assumed was a clinically relevant environmental condition. What I mean by clinically relevant is something that in humans would predict vulnerability to illness. And so what we chose was what we consider to be the preeminent risk factor or social determinant, mom, right? Everybody's got one. Right? And they predict health outcomes in many different spheres, including well beyond mental health. So we wanted to ask whether or not variations in maternal care could produce sustained changes in gene transcription and downstream changes in function. So we needed a model to do that. And these are, what you're looking at is our model. These are Long-Evans Norway rats. And it may surprise you to know that these mothers actually show very stable individual differences in maternal care in their treatment of their offspring. So the behavior that distinguishes these mothers is this licking and grooming behavior that you see pictorially represented here. So in the course of a nursing bout, mothers lick their pups. And people like Myron Hoffer and Saul Schanberg and Gig Levine had shown how this licking dramatically alters the endocrine systems in the developing animal. For example, pituitary growth hormone release is governed by the mother's licking of the pup. Tiffany Fields translated that into a common NICU intervention program. So the pup's physiology is being dynamically regulated by this licking. And moms show these stable individual differences. And they're stable not only across days within a litter, but even across litter. If a mother's a high-licking mother, she'll be a high-licking mother basically throughout the course of her reproductive life. Same is true for low-licking mothers. And we just did what any developmentalist would do. We simply asked the question, if we statistically define high and low-licking mothers, can we then follow these animals through developmentally and look at the correlates at the level of phenotype? And we did that with respect to, and have done that with respect to, behavioral and endocrine responses to stress, neural development, particularly looking at hippocampal and prefrontal and amygdaloid systems. We do that in many forms of learning and memory. We do that in terms of metabolism. By the time of midlife, particularly the female, but also the male offspring of low-licking mothers become virtually fully metabolic. We look at reproductive outcomes, and I'll show you a brief example of that, but one of them, for example, is that the female offspring of low-licking mothers go through puberty about 4 to 5 days earlier than do those reared by high-licking mothers. So there's these stable variations in phenotype, and then the question becomes, well, can you move beyond this statistical correlation? And of course, the way we do that, we just use a classic cross-fostering paradigm. We take the biological offspring of lows, foster them onto highs. We take highs, foster them onto lows. And with respect to these phenotypic outcomes, every one of them is either partly or in certain cases completely reversed with cross-fostering. And so for that reason and others I'll show you, we have come to believe that indeed mother's licking is driving this phenotypic variation that's occurring over the course of postnatal development. So now we've got a mechanism, we can start to understand or potentially address the question of how it is that parental care may give rise to these epigenetic marks, leading to changes in gene expression and changes in more complex phenotype. And the primary phenotype of interest is that of the Hypothalamic-Pituitary-Adrenal System. It's a system I'm sure you know very, very well. Its nexus, of course, is a population of neurons in the paraventricular nucleus that make their living by synthesizing a peptide, corticotropin-releasing factor, and releasing it in response to anything stressful. It activates pituitary and adrenal release, including the release of glucocorticoids. There is a negative feedback loop critically to this, where glucocorticoids act at glucocorticoid receptors within the hippocampus, and the net effect of that interaction is to diminish the synthesis of CRF and to moderate subsequent pituitary adrenal responses to stress. And in a nutshell, what you find if you look at the offspring of high and low licking and grooming mothers is that animals reared by high licking mothers show an increase in glucocorticoid receptor expression in the hippocampus by compared to those reared by low licking mothers. The high licking offspring show subsequently increased they show not only increased glucocorticoid receptor expression, predictably they also show increased feedback sensitivity, so they can actually more effectively inhibit CRF in response to elevated circulating levels of glucocorticoids. And this effect is to associate with decreased levels of hypothalamic CRF, and not surprisingly then when you come in and stress these animals, there's a greater response to stress in the offspring of low compared to high licking animals. And that, by the way, can be reversed by using either pharmacological or genetic treatments that eliminate the influence of the hippocampal glucocorticoid receptor. So the net effect there was that we're able to at least identify a gene that we know is actively regulated by maternal care and that is involved in regulating this difference in the stress response. So the question then became that of trying to actually understand how it is that this difference occurs. And what we did was to first of all go back and start to ask the question about the differences in the regulatory region that control the expression of the glucocorticoid receptor. We identified a sequence of DNA that was responsible for producing the glucocorticoid receptor in the hippocampus, and what we found was that animals raised by high-licking mothers showed an increase in the methylation, decrease in the methylation, of the promoter region that associates with glucocorticoids by comparison to those reared by low-licking mothers. So the bottom line essentially was that animals reared by low-licking mothers had increased methylation of the promoter region and decreased glucocorticoid receptor expression, and that was essentially what was associated with the difference in glucocorticoid receptor expression. Now, I mentioned to you earlier that differences in methylation are associated with downstream signaling, such as this histone deacetylase, which then comes to assure a closed configuration and silence to transcription. So, for example, the increased presence of a histone deacetylase associates with decreased histone acetylation and decreased capacity of transcription factors to bind to the site and to turn on the glucocorticoid receptor. So we had defined, for example, the difference in methylation. The next question we did was to then ask whether that difference in methylation was showing, as predicted, associated with a change in histone acetylation. What you would predict, of course, is that increased methylation attracts the histone deacetylase and associates with decreased histone acetylation, and that's exactly what we found. So if you look, this is brain tissue taken from the hippocampus of animals reared by high and low licking and grooming mothers. We're looking at that regulatory region that guides the activity of the glucocorticoid receptor, and you can see that there's increased acetylation of that region of the DNA in animals reared by high compared to low licking mothers, which reflects open chromatin and active transcription. We looked at another mark, in this case a mark on histones that involves methylation, that is also associated with active transcription. And once again, in the adult offspring of high compared to low animals, we find increased histone modifications, again actively associated with transcription. So bottom line then is the increased methylation of this glucocorticoid receptor promoter in animals reared by low licking mothers is associated with decreased histone acetylation, closed configuration of chromatin, decreased capacity of the transcription factor NGFIA that regulates glucocorticoid receptor expression, and thus decreased expression. So this is the chain of event as we imagined it. Now, what we then did was to ask that question: if indeed this process, this epigenetic process, is critical for the difference both in gene transcription as well as in pituitary-adrenal function. Now, if indeed the difference in methylation is mediated by a histone deacetylase, what we should be able to do is to take adult animals reared by high and low licking and grooming mothers, infuse them with a compound that disrupts the capacity of histone deacetylase to act, and thus reverse the phenotype. So that's what we did. We've got adult animals raised by high and low licking grooming mothers. We infused trichostatin A, a broad spectrum histone deacetylase, and what we do is to completely reverse the difference in histone acetylation We reverse the difference in transcription factor binding. We reverse the difference in glucocorticoid receptor expression. And most importantly, we also reverse the difference in pituitary-adrenal responses to stress. So the animals that are raised by low-licking mothers, but infused into the hippocampus with a control vehicle, they show an exaggerated glucocorticoid response to stress by comparison to animals that are reared by high-licking mothers. These are the animals in the red or the blue. Conversely, animals reared by low-licking mothers, but infused with the histone deacetylase, not only do we reverse the differences in effects on glucocorticoid receptors, you also reverse the difference in the pituitary-adrenal response to stress. So this provides some evidence that these epigenetic variations that are linked to maternal care are actually guiding the differences in glucocorticoid transcription as well as the difference in pituitary-adrenal activity. We've gone on over the course of about, goodness knows, about 15 years to actively describe, yeah, it takes a while, to describe how it is that maternal licking actually comes to produce these differences. The tactile stimulation of maternal licking activates pituitary thyroid systems. What it does so actually is indirectly. What it does is to activate noradrenergic signals, which then lead to the monodeiodination of thyroxine 2-triiodidithyronate, T3. This is the most biologically active form of the thyroid. Thyroid hormones, of course, have pervasive effects on the growth throughout the body. One of their major effects is on ascending serotonin systems. So the increase in licking and T3 is associated with an increase in serotonergic signaling at the level of the hippocampal neuron. And this ultimately translates into changes in intracellular pathways, the activation of a transcription factor, NGFIA, as well as SP1, a cofactor, the Krebs binding protein. And collectively, we now know that these signals, which are induced by maternal licking, are absolutely obligatory for the change in methylation. So these are the pathways by which a social event, essentially maternal licking, is being translated into a change at the level of the epigenome. And we can actually show that the binding, this binding of NGFIA to this critical promoter region, this regulatory region to the glucocorticoid receptor, is directly associated to maternal licking. So Ian Hellstrom in the laboratory performed an experiment in which he took tissue from pups at postnatal day 4 and excised the hippocampus, and that tissue was taken immediately after a nursing bout that included licking or grooming, or it was taken 25 minutes later. So what happens is these mother rats are showing nursing bouts that generally last for about 20 to 25 minutes, and then they take a break for about 25 to 30 minutes. It's after the break or directly after the nursing that Ian collected his tissue. First of all, what he's then doing is looking at the binding of this transcription factor directly onto the DNA, and he sees that, for example, maternal care does indeed associate with increased interaction between this transcription factor and the DNA, but that occurs only following a licking period. The off period, for example, the highs are indistinguishable from the normal high, or if the lows are indistinguishable from the offspring of high licking mothers, so that the binding, the interaction that we refer to here of NGFIA with the DNA is in fact being actively regulated by maternal care. That's occurring not only with respect to the glucocorticoid receptor, there are other genes that are being regulated by the same transcription factor NGFIA, and that those genes, the interaction of NGFIA with those DNA regulatory sequences similarly being regulated by maternal care. And Tiwan Zhang in the lab has shown that if you look at these other genes, that they show the same pattern. The GAD1, the glutamic acid decarboxylase gene, is also regulated by NGFIA, and it too shows a difference in methylation that is sustained into adulthood, and that it is following pretty much the same pattern. So it's reasonable to assume that these social events are altering intracellular signals that are then altering the binding of specific transcription factors to the DNA, and thus creating an opportunity for remodeling these epigenetic signals over time. So this suggests that, as I said, that maternal care is indeed activating intracellular signaling systems. There's no big surprise to that. What's interesting is that the activation of these signals can, in fact, create a situation in which there's the possibility of remodeling epigenetic marks at particular sites of the DNA. One of the interesting things, and I'll just show this for people who may be aficionados of epigenetics, is that this story began with certainly the study of methylation. In the last few years, it's become clear that methylation exists in multiple forms. There are many flavors of it, and that unfortunately, the original techniques that we used cannot distinguish between what are the primary forms, which is 5-methylcytosine. 5-methylcytosine can be further processed into 5-hydroxymethylcytosine with an TET1 enzyme. And as I say, we then don't know exactly what form this methylation exists. We can study it, though. And Tiwan Zhang in the lab has developed the methodology using TET-assisted sequencing. And we can sequence across the entire genome and ask whether or not this signal exists in the form of 5-hydroxymethylation or 5-methylation. This is the area of interest. It's the promoter region to the glucocorticoid receptor. And what you're looking at is just next-generation sequencing across the entire glucocorticoid receptor gene. And what you can see is that this is the tab sequence. It's this track that identifies hydroxymethylation. It's this track down here that identifies the classic 5-methyl-C. And if you can see, can you? I haven't actually checked this, but I should, is that it's largely in the form of 5-hydroxymethylation that you see that signal. And indeed, when we go out and use techniques that can distinguish between the two, we find that it's existing in this particular form. Now, to most of you, you can actually write this particular part of the talk off completely. The reason I'm raising this is just to caution you that there's a really moving target in the study of epigenetics, and in particular trying to interpret what these particular signals mean. 5-hydroxymethylation can take on a very different life form and can have very different meanings. One of the interesting features of it is it's much more readily modifiable than is the classic 5-methyl-C. So it's interesting that potentially these environmentally induced signals of methylation exist in what could be a more transient form of methylation, suggesting reversibility. But as I say, that's basically where we go. We've started to look at a range of other phenotypes and started to ask whether or not the same processes might apply to these particular outcomes. We've looked at synaptic plasticity. One of the major genes that comes up when we survey the transcriptome, we look particularly at the hippocampus, and studies using long-term depression, long-term potentiation, is that we find these perisynaptic mGluR1 receptors differ in their expression as a function of maternal care. You get increased expression of the mGluR1 receptor in adult animals raised by high compared to low-licking mothers. That associates with changes in the potential for synaptic plasticity, particularly in the form of long-term depression. And the mGluR1 gene, as Rose Baggett and Tiwan Zhang showed in the lab, is epigenetically regulated. It's regulated by maternally induced changes in methylation. So we've now started to work through a number of these phenotypic outcomes and associate them with what I think are epigenetic signatures. The one I'd like to talk to just briefly about is one that I think is absolutely critical in terms of thinking about this in a transgenerational context. And that's really the work, first of all, of Frances Champagne when she was a graduate student in the lab, and more recently Dara Chirac. And that's on the issue of looking at the transgenerational transmission of the individual differences in maternal care that we think launch this particular variation in phenotype. So the first thing we did was to actually start to look at this by looking at the female offspring and found that that great female rat nightmare comes true. They become their mothers. And so indeed, females reared by low-licking mothers, on the whole, show a reduced frequency of licking compared to females that are raised by high-licking mothers. And Frances did an extraordinarily detailed cross-fostering study to show that indeed that was transmitted through maternal care in the postnatal time of development. And then she and Darlene Francis had previously shown that what seems to be at least one of the core neuroendocrine mechanisms for this is that the adult virgin females raised by high-licking mothers show increased estrogen receptor alpha expression in the medial preoptic area compared to those reared by low-licking mothers. And many scientists dating back to Jay Rosenblatt's work in Newark had shown that the activation of estrogen receptors in the medial preoptic area is absolutely obligatory for the female rat to become maternal. And so here what we're seeing is that that mechanism seems to exist at a different level, and Francis went on to show differential sensitivity in the medial preoptic area to estrogens. And what happens is that estrogen receptor alpha enhances oxytocin receptor binding, which has a feed-forward effect on oxytocin neurons. You end up with greater oxytocin stimulation of dopamine, and Dara went on to show that that's actually permissive to the differences in the licking and grooming in the offspring of high and low licking and grooming mothers. So it's an estrogen-induced effect, downstream of which we see oxytocin-dopamine interactions. Francis went on to show that the difference in estrogen receptor expression in the medial preoptic area can also be reversed in cross-fostering. So here's the difference in ER-alpha expression in the MPOA in high versus lows. Animals that are born to highs but reared by lows show diminished expression. Animals, conversely, that are born to lows but cross-fostered onto highs show increased expression. So it's regulated by maternal licking. And Francis went on to show that if you take the promoter region that we know regulates estrogen receptor expression in the medial preoptic area, that it's differentially methylated. So here what you see is just differential methylation of individual cytosines across this exon 1B promoter that regulates ER-alpha expression, and you see increased methylation in tissue that's taken from high compared to low-licking mothers. And the methylation exists in regions that actually bind a transcription factor, STAT5, that normally turns on ER-alpha. So in fact, what Francis and Dara and others have shown, Sabine Deer included, is that this methylation associates with decreased transcription factor binding and thus decreased expression. Interestingly, what I think Sabine Deer has shown is that in fact you can link the differences in ER-alpha expression to, first of all, the expression of changes in maternal care. And so what she did was to simply take animals who are the offspring of high or low licking grooming mothers. So we would expect, as we see here in the control animals, that the offspring of mothers who are high licking mothers would show increased licking and grooming compared to those that are raised by low licking mothers. What she then assumed was that if she essentially neutralized the capacity or diminished the capacity in the medial preoptic area to express the ER-alpha gene, so she simply induces an shRNA that essentially diminishes the active transcription of the estrogen receptor alpha gene, is that she completely eliminates that particular difference. So it is ER-alpha and the differences in expression in ER-alpha that is regulating these subsequent downstream effects on maternal care. And what she then showed also predictably is if you take the female offspring of these animals, in the absence of any further manipulation, is that the offspring of these animals, which have a lineage of high licking and grooming, actually come to resemble low licking mothers. So that it's the behavior of the mother that is actually guiding the transgenerational transmission. So we've come to then think of DNA methylation as a signal that may potentially then stably imprint the social experience of the animal onto the genome, and thus may guide variation in phenotype and potentially health outcomes. So we started to then ask whether or not we could translate these particular findings in any meaningful way into the humans, and that's what I'd like to take you through for the rest of the talk. Our first foray into this particular zone involved a collaboration with a good collaborator and friend, Dr. Gustavo Turecki at McGill. Gustavo runs the Quebec Suicide Brain Bank, which, as the name implies, is a repository from tissues who are suicide victims or controls. This allows us, of course, to go and excise exactly the tissue of interest, the hippocampus. Gustavo has also masterminded the development of a clinically validated forensic phenotyping. So we can not only look at the right tissue, but we can then understand whether these individuals had a current or previous history of psychopathology, as well as broad features of their developmental history, which, as you'll see, comes to be critical. And we can look at the same region of the gene. So in the human, the glucocorticoid receptor gene's organized in very much the same way as it is in the rat. If you look at that area that's an ortholog, it's this exon 1F. That's the promoter that guides the expression of the glucocorticoid receptor in neurons that is comparable to the promoter that we looked at in the rats. And when we then just took this brain tissue and then just did simple QRT-PCR, what you find is that in tissue samples, hippocampal samples taken from controls, there's an increase in total glucocorticoid receptor messenger RNA compared to suicide tissues, as well as an increase in transcripts that bear this exon 1F signature. So this promoter appears to be more actively regulating transcription and potentially thus accounting for the differences in messenger RNA for the glucocorticoid receptor. Well, we then asked the question, does this difference map on to changes that we see in psychopathology? So we had enough samples to do meaningful correlations with either major depressive disorders or with drug abuse, particularly alcohol and psychostimulant abuse. And we found nothing, no correlation whatsoever between the expression of the glucocorticoid receptor and these forms of psychopathology, either currently or previously, nor did it associate with any drugs that were on board at this particular point in time. There was nothing we could do to explain it. So we started to look then at the developmental history of these individuals, and what we had was a sufficient number of suicide samples, 24, which literally half were positive for a verified history of childhood abuse or maltreatment, and half that were not. And as you can see, the difference in glucocorticoid receptor expression is entirely unique to those individuals with a history of abuse. It doesn't appear at all amongst individuals who were suicide completers in the absence of a developmental history of abuse. The same is true for this particular exon 1F promoter, and that's the promoter, as you'll recall, that's regulating glucocorticoid receptor expression. And so what we then did was to ask about the methylation status of this promoter and found that indeed, not surprisingly, it's hypermethylated in individuals with a history of abuse. And that indeed the presence of methyl groups in these specific regions blocks transcription factor binding and transcriptional activation. So it was a story that was at least reminiscent of what we'd see in the rat. It was also consistent with what had been previously published in studies of the sequelae of abuse. These are studies by people like Christopher Lee and Christine Hyman and Michael DeBellis, all of which had shown that maltreatment associates with an increased expression of CRF in the hypothalamus and a greater capacity for ACTH and glucocorticoid responses to stress. And that, of course, would be associated with decreased glucocorticoid receptor expression in the hippocampus. So that story appeared to be of some value. Now, the interesting thing, of course, was that tissue was obviously of brain origin, neural origin. So we were fortunate enough to be able to acquire postmortem tissue. As it turns out, a number of other studies, in fact, we've just completed a meta-analysis, and there are now 14 studies that have attempted replication of this particular finding, that is, looking at the relationship between social adversity during perinatal development and the methylation status of the glucocorticoid receptor, for the most part looking at that same promoter region. And 13 of those studies have actually replicated this particular effect, and one failure to replicate. So even though these studies are using blood or in some cases buccal epithelial cells, they continue to see this particular association. Well, the interesting part of that is that that means that we might be able to pose interesting questions about the epigenome without necessarily being able or obligated to access human brain tissue. And I think there's now emerging evidence that indeed that actually may be a fruitful endeavor. This is some work by our collaborator, Michael Kobar, at the University of British Columbia. What Michael was doing was to acquire DNA from buccal epithelial cells from individuals in which he was aware of and had been measured early socioeconomic versus current socioeconomic status, total cortisol levels, and total levels of stress. And as you can see, when he looks at variation, he's using a genome-wide analysis that I'll tell you about in a few minutes. When he looks at variation in methylation across the entire genome, he can find a significant influence of early but not current socioeconomic status, and he finds an influence of cortisol as well as stress. So in essence then, what I think these studies tell us is that we can start to interrogate the epigenome broadly. Now, we're not necessarily going to find individual epigenetic mutations necessarily that correlate with phenotypes. We can start to ask questions about the epigenome and how those variations in the epigenome are defined. And that's the nature of the type of studies that we've been doing recently. So, for example, one of the studies has looked at a cohort study. This is a birth cohort study based in Montreal and in Hamilton, Ontario, in which we've been able to take samples from kids who are 6 to 8 years of age. So we can take buccal samples, these are just cheek swabs, extract the epithelial cells and the genomic DNA, and then to ask whether we can then look at variation in methylation across these particular samples. The tool we use is called the Infinium Human Methylation 450Ka bead chip, and what it does is it takes purified genomic DNA and allows you to interrogate the methylation status of 488,000 individual CPGs, which sounds like it'd be fabulous, right? Except it turns out to be like 3% of all the CPGs across the genome. And you take what you can get. So we get a number, to be sure. And what it gives you, though, across any particular genome is a fairly good picture of global changes in methylation. This is just one particular gene. This is a MAP kinase. It's got a really good coverage. It's covered by about 34 sites. Here you can see the regulatory region of the gene. This is known as the 5' untranslated region, upstairs from the transcriptional start site. As you can see, this gene shows very low levels of methylation in the regulatory area. In the gene body, conversely, it shows hypermethylation. Remember I told you methylation can have different effects? Well, methylation in the gene body enhances transcription. Here it has exactly the opposite, more traditional. So this gene is basically showing you the signature of an actively transcribed gene. It's got low levels of methylation here and high levels of methylation there. More importantly, what it does is allows us to quantify that across these areas. And so one of the things we've been doing is to actually look at whether or not we can look at variation in the methylome in relation to things that developmental psychologists would understand as being predictive of later outcomes. And so we looked at the Ainsworth strange situation test. So at 36 months of age, the kids went through the strange situation test. They were then obviously categorized as secure, insecure, or disorganized. And we then took the methylation data and asked whether or not there was variation across the genome here. We used principal component analysis and found that there was a significant component, PC2, that accounted for 7% of the variance across the epigenome, and that principal component itself was associated with childhood attachment at 36 months of age. And that the effect is primarily, as you can see, between disorganized versus secure and insecure forms of attachment. So attachment at 36 months of age was statistically associated with variation in the epigenome at 6 to 8 years of age. And importantly, Kieran O'Donnell, who led these studies, showed that that's a stable effect. So when he came back from the same kids a year later, he showed that that difference was there. In fact, the correlation across different times in the same subject is 0.9981, so it's fairly reliable over time. So that the effects that are occurring here are likely to be stable, at least across middle childhood. We've started to look at other components of this signal. And really here we've answered a question, I forget, we were talking, Adam, earlier about this particular. We've started to ask the question about whether we can study epigenome as we do other elements of phenotype and start to understand their emerging definition as a function of both genetic and environmental effects and, of course, their interaction. So these studies have been done using the same human methylome chip. We have 200, well, at the time we did the study, we had 237 samples. This is in a birth cohort study in Singapore. We have full genotyping using the Human Omni Express bead chip, and we have multiple measures of maternal conditions prior to birth. So obviously umbilical cord tissue, we're getting this at birth. And then we start to ask the question, well, we can start to then look at the degree to which variation in the epigenome is associated with genotypic versus environmental or their interactive type of processes. So the way we did this statistically, by the way, is certainly by proxy and independent. We're using principal component analysis to analyze these data. What we did actually was to take the original data and start to ask about variation across these particular regions. So first of all, we've got 488,000 CPGs, and we started to ask, well, what percentage of those CPGs show any variation from one individual to another? As it turns out, it's about a third, right? And that estimate, by the way, is going to vary depending on what tool you use. But it's important to understand that about 70%, at least, if not more, of all the methylation on any DNA is invariant across individuals, right? There's regions of the genome that we do not want expressed. Thank you. Most of it of viral origin, so we methylate the hell out of it. We do it in every cell in the body, and it's invariant. And that's a property of life. If we don't go on. So a lot of methylation is just not going to vary. So what we want to do, of course, is to focus on that part that does. Joanna Holbrook, who led this analysis, defined this variably methylated region as a region of about a kilobase, 1,000 base pairs, in which there was variation in methylation in at least 2 or more CPGs. So when we do that, we find 1,423 regions of the genome that show variable methylation. We then take the OmniExpress and about the 1.4 million polymorphisms that it finds. We then hand that over to the computer science, the computer resources, and we take 2 weeks of computing time because we want to ask which of these 1.6 million SNPs best correlates with each one of these 1,423 variably methylated regions. So plug it in and then wait 2 weeks. And what you get for every one of these 1,423 regions is the SNP, the polymorphism, that is most statistically associated. That's what we're calling our genetic influence. It doesn't mean there are other SNPs associated with it. It's just our best proxy for a genetic influence. So we find a considerable genetic influence. Now, by way of Pca analysis, one of the things that's really interesting to look at, this is the Pca analysis of the SNPs, the genotypes themselves. So these are the 3 populations in Singapore: ethnic Chinese, ethnic Malays, ethnic Indians. And as you can see, a Pca analysis can resolve these genetic polymorphisms very effectively. Not so much the epigenetic differences, right? So here you're clearly seeing variation between genotype and phenotype, in this case at the level of the methylome. So now we're asking what defines that? Well, the first thing that we've got is we've got SNPs. We've got a SNP associated with each one of our 1,423 variably methylated regions. Now, some of these are really boring. All the SNP is doing is taking a C and changing it into an A. A's can't be methylated, so duh, you don't get methylation. As it turns out, that only accounts for 12 of all of these events. Much more likely to occur is that the SNP is here and the change in methylation is in close proximity, about 100 base pairs. So you're seeing genetic and epigenetic differences in close proximity. Then we start to get serious and say, okay, here's what we want to do. We want to take all of these 1,423 variably methylated regions and ask, is the variability across that region best associated with genetic influence, environmental influence, or gene by environmental? Now, we're not saying only by any means. We're just saying which one of these linear regression models best explains the variation in the methylation. And what you find across the whole population is that for 25% of these particular variably methylated regions, it's the genetic influence that best explains that effect. For 75%, it's a gene by environment interaction. And for 0%, is it the environment? And then, of course, somebody points out cleverly that we've got a mixed ethnic background. That's going to enhance the prediction of the genetics. So we say, fine, we'll just take the Chinese, ethnic Chinese alone. And this drops down to about 16%. So within any specific ethnic population, at least using umbilical cords, our estimate is that somewhere between 15 and 20 some odd % is best explained by that SNP. The remainder is best explained, most of it's best explained by gene-environment interaction. So then we start to ask the question, well, okay, what would that actually look like? And so we drilled down on what, for many of us, is a very common variant that's been very well studied. This is the Val-Met polymorphism in BDNF. And it's been rumored to be associated with plasticity of phenotype, and of course BDNF is a trophic factor. It regulates synaptic plasticity throughout the nervous system. And so we started to say, okay, well, let's look at the BDNF gene and use it as a poster. Well, let's look at it as one of those, if you will, gene or potentially gene by environment interactions. So we identified regions of the BDNF gene that were associated with changes in BDNF. But what we were most interested in doing was to drill down on one of these gene by environment interactions. And so what we did was to look at maternal mental health. We looked at maternal depression and maternal anxiety. And we asked the question, since both maternal anxiety and maternal depression interact with genotype to affect a significant percentage of the methylome, what would that look like with respect to this BDNF SNP? We know that the various SNP, for example, the MET-METs differ from VAL-VALs. It is, at least within Asian populations, suggested that MET-METs show increased predisposition for anxiety. Francis Lee at the other Cornell campus has humanized a mouse with this particular variant and shown that MET-MET mice appear to be more anxious. So we asked the question, could the BDNF gene be influencing the influence of antenatal maternal anxiety on methylation? And so here's what you find. Looking here, for example, on the ordinate is just a simple number of brute CPGs that are variable, right? And what you can see is that as a correlate of either state or trait anxiety, many more CPGs are affected by anxiety in children, infants carrying the MET-MET versus the VAL-VAL variant, right? So clearly a much greater effect of antenatal maternal anxiety in MET-METs compared to VAL-VALs. True of state, true of trait. That's modified by infant genotype, not by maternal genotype. This is maternal Val-Met polymorphism, and as you can see, there's nothing that easily maps onto that what we see in infants. So it's being regulated by the infant's BDNF MET. So yes, here's an example then of an interaction between a specific SNP and an antenatal condition and its impact on the epigenome. So then we took advantage of the fact that Chuan Ji and her team in Singapore have been imaging these same kids at about the same time. So this imaging of the kids is done between 4 and 10 days postnatal. So it's pretty much about the time that we're doing our epigenetic survey. So she then asks, if I take these variably methylated regions that are associated with antenatal maternal anxiety and map them on to the brain, what do I see? Now, she looked at 19 different regions, and in most of these regions, there was nothing that occurred that wasn't eliminated by correcting for total brain volume. The two most affected regions that were independent of total brain volume and as well as of gender, etc., were the right amygdala and the left hippocampus. Okay? Now, in the case of the right amygdala, what she found was that there was about 3 times as many CPGs in the MET-MET group that correlated with volume in the right amygdala than in the MET-VAL or VAL-VAL. So those CPGs that are being affected by antenatal maternal anxiety are more closely associated with the right amygdala in individuals who are carrying that MET-MET polymorphism. If you look at the hippocampus, it's exactly the opposite. In this particular case, CPGs that associate with maternal anxiety are actually in the VAL-VAL group associated with the left hippocampus. So what's happening here defies any simple notion that the VAL-VALs or the MET-METs are more sensitive to antenatal maternal anxiety. Instead, what it's actually saying is that what the polymorphism is doing is, first of all, determining which CPG sites are affected by antenatal maternal anxiety. It's obviously influencing the number of them. But perhaps even more importantly, it's also determining what the impact is potentially at the level of the brain. So I think what it does is it takes us to understanding gene-environment interactions beyond the simple level of saying, are VAL-VALs more sensitive than MET-METs, etc. It obviously determines what's the outcome. So I'm going to take you through, this talk, by the way, does come to an end at some point in time. I'm sorry, but as the day goes on and you talk to people, you say, oh, I'll throw in that slide. No, I'll throw in that slide. No, I'll go on lithium. I'll take you to one last topic, and that is a partnership that we've developed, which is a cherished partnership. This is with David Olds and the group who run the Nurse Family Partnership Study. Many of you will be at least as familiar, if not more so, with this program than I. It started just down the road here, right? In Elmira. Yep. Exactly. That's not hard to imagine at all. So this is a partnership that involves the nurse and family. It involves nurses trained to work with high-risk mothers. These are largely mothers who are at risk for abusing their children through all the risk factors that many of you would know all too well. The interactions, the treatments involve prenatal and postnatal, extended visits from the nurse to the mother and her child, but those visits extend only to 24 months. And so what we were able to do was to acquire blood samples from the Elmira group. Now, the data I'm showing you is from 90 subjects. That's now is 279. And so the data remains very much the same. So we've got blood samples from which we extract DNA at 27 years of age. So we can then ask, and again, we're not after individual CPGs, we're simply saying, can we look at the methylome of these individuals 25 years after the intervention has occurred and detect a signal that statistically associates with the presence of that intervention? That's what we're trying to do. So we're using the same tool, this 450K, and what we're able to find is that when you look across the methylome, you see predictably a change of gender. Males and females differ in methylation quite robustly, by the way. And that's true of all tissues that I know of. We see a signal that associates with abuse up to 4 years of age, and an even stronger signal, and that's emerging actually as we continue the statistical analysis. Abuse up to 15 years of age is a very potent predictor. But what you also see is a treatment effect. So 27 years later, you can come back and identify variation in the methylome that is associated with this particular treatment effect. And importantly, the treatment effect itself, the PC, the principal component that is associated with the treatment, itself predicts specific treatment outcomes. Again, these are not overwhelming effects, but the fact that you can come back 27 years later, 25 years later and identify. So the principal component that associates with treatment predicts substance abuse and disorders. So some idea that these effects can, first of all, be associated with intervention programs, that those effects can be enduring, and that they can associate with functional outcomes. So just by way of a brief summary, pretty much what we've gone through. I think it's reasonable to think that variation in the epigenome, and that is those biochemical signals that regulate structure and function of the genome, that they are subject to environmental regulation. That's probably true over the course of the lifespan. Our data to date are most focused in terms of developmental events. But importantly, these epigenetic signals, to some extent at least, reflect the quality of the early environment, and they do and are functional in terms of guiding sequence transcriptional activity within the brain. And it's realistic, I think, to start thinking of the epigenome like any measure of phenotype, that it's going to be the product of constant gene-environment interactions and needs to be studied as such in that particular context. Thank you very much.